Imagine a world where your blood type no longer decides whether you live or die while waiting for a kidney. For decades, that biological lottery has shaped transplant medicine. Type O patients stuck at the back of the line, watching others with more compatible blood receive life-saving organs while they wait, sometimes for years. But now, science has taken a bold leap toward rewriting those rules. In a groundbreaking experiment, researchers have successfully transformed a kidney from one blood type into a universal donor organ, then implanted it into a human body to see if it could survive. The result? A functioning kidney that fooled the immune system, for a crucial window of time, and opened the door to a future where any donated organ might work for anyone. This is not science fiction. It is the next chapter in a quiet revolution happening inside labs and operating rooms, one that could dismantle one of the oldest barriers in transplant medicine. The key lies not in gene editing or futuristic machines, but in a clever trick using nature's own tools, enzymes. These tiny proteins act like molecular scissors, precisely snipping away the chemical tags on the surface of cells that define blood type. Every red blood cell, and every organ, carries these tags, known as antigens. Type A blood has A antigens, Type B has B, AB has both, and Type O has neither. That absence is why Type O is considered universal. It lacks the markers that trigger immune alarms in other people's bodies. But what if you could strip those markers off a Type A organ, making it look like Type O? That is exactly what scientists have now done. The process begins with an organ that would otherwise never be used, perhaps damaged, or mismatched, or from a donor whose blood type doesn't align with any waiting recipient. Instead of discarding it, the organ is connected to a perfusion machine, a device that mimics blood flow by circulating a nutrient-rich solution through its vessels. Into this fluid, researchers add specially engineered enzymes. These enzymes seek out and remove the A antigens from the surface of the kidney's cells. Within a couple of hours, the organ's biological identity is effectively rewritten. On paper, it was Type A. Now, to the immune system, it appears as Type O, a blank slate, ready for any recipient. To test whether this illusion would hold, the team turned to a unique and ethically sensitive model, a brain-dead individual whose body was still being maintained on life support, with full consent from the family. This person had Type O blood and, critically, high levels of antibodies primed to attack anything with A antigens, the worst possible scenario for rejection. Normally, transplanting a Type A kidney into such a person would cause immediate, catastrophic failure. But this was no ordinary kidney. After implantation, something remarkable happened. The kidney began working. It filtered waste, produced urine, and showed no signs of distress. For 2 full days, it operated as if it belonged. Then, on the 3rd day, the spell began to break. The kidney's own cells started regenerating the A antigens that had been stripped away. The immune system, ever vigilant, detected the returning markers and launched an attack. Rejection set in. But here is the crucial insight: those 2 days of peaceful function were not just a fluke, they were proof that the conversion worked. More importantly, in a real transplant scenario, doctors do not leave the immune system unchecked. Patients receive powerful drugs to suppress immune responses, along with treatments that remove or neutralize harmful antibodies before surgery. If those standard protocols were combined with this enzyme conversion, the window of tolerance could stretch from days into weeks, months, or even years. This matters because the organ shortage crisis is not just about numbers, it is about compatibility. Thousands of kidneys are discarded each year not because they are damaged, but because they do not match the blood type of available recipients. Meanwhile, type O patients languish on waiting lists, their chances of survival dwindling with every passing month. By turning type A or B organs into universal ones, the pool of usable kidneys could expand dramatically. Suddenly, a kidney from a type A donor in California could save the life of a type O patient in New York, without the agonizing wait or the complex logistics of paired exchanges. The implications ripple beyond kidneys. The same enzyme technique has already shown promise in lungs. Hearts, livers, and pancreases may follow. The dream is a single, streamlined process that could be applied to any solid organ during the standard preservation period between donation and transplant. No extra surgery, no genetic overhaul, just a few hours of perfusion with the right enzyme cocktail. It is elegant in its simplicity, yet profound in its potential. Of course, challenges remain. The antigen removal is temporary because the organ's cells continue to produce the original blood type markers over time. That means the converted organ is not permanently universal, it is universally compatible only for a limited period. But that may be enough. The most dangerous phase of rejection happens in the first days after transplant, when pre-existing antibodies can destroy the organ almost instantly. If the enzyme treatment can buy enough time for immunosuppressive drugs to take hold and for the body to begin accepting the new organ, long-term survival becomes possible. Ethically, the use of brain-dead recipients as test subjects raises questions, but it also offers a rare and valuable middle ground. These are not living patients risking their lives in an unproven trial, nor are they animals whose biology may not mirror human responses. Brain-dead bodies, maintained with family consent, provide a human testing ground that bridges the gap between lab dishes and clinical reality. They allow scientists to observe how a real human immune system reacts to a modified organ in real time, information that cannot be gained any other way. What makes this advance so compelling is that it does not require reinventing transplant medicine. It works within the existing framework, using tools already in hospitals, protocols already in place, and organs already being recovered. It is an upgrade, not a revolution. And because it builds on decades of research into blood group biochemistry and enzyme engineering, it is grounded in solid science, not hype. Looking ahead, the path to widespread use will involve refining the enzymes for greater efficiency, ensuring complete antigen removal, and testing the approach in living recipients. Regulatory approval will take time, as it should. But the foundation has been laid. For the first time, we have direct evidence that a human organ can be chemically reprogrammed to evade immune detection, at least long enough to matter. In a world where medical breakthroughs often feel distant or abstract, this one is tangible. It speaks to a future where biology is not destiny, where the accidents of blood type no longer dictate who gets a second chance at life. It is not a cure-all, but it is a powerful new tool, one that could shorten waiting lists, reduce organ waste, and most importantly, give hope to those who have waited the longest. The kidney that worked for 2 days in a brain-dead body may one day be the prototype for thousands that beat, filter, and sustain life in people who once had no match. And that is a future worth fighting for. If this breakthrough gives you hope, or just blows your mind, hit that like button and subscribe for more stories where science meets real life. Drop a comment below. would you trust an enzyme-converted organ if you ever needed a transplant? And remember, the future of medicine isn't just being written in labs. It's being built, one reprogrammed organ at a time. Thanks for watching.